Hi everyone, this is the second part of Lecture 4, the Journal Club, on the article entitled Value-Added Biotransformation of Cellulosic Sugars by Engineered Saccharomyces cerevisiae. In this part, we are going to look more at the products that can be produced by cerevisiae. We said that Saccharomyces cerevisiae is a very good ethnologen. It makes very high rates and titers of ethanol that can be used as a biofuel. But we will now explore some other products that can be produced in yeast that may be of interest in a biorefinery concept. Some of these are produced at high levels and are actually in commercial production already, whereas others are still under development, and we'll see the production levels are quite low, and it's more of at a proof of concept stage. So the first product we're going to look at is xylitol. Now, in addition to ethanol, engineered yeasts have been used in the bioconversion of xylose to a variety of other alcohols. One example is that sugar alcohol xylitol. That is the first intermediate in the oxidoreductive xylose consumption pathway. And I said this can accumulate in the utilization of xylose, and it's actually a negative factor there. But xylitol is an interesting product on its own. It's used in the food industry as a low calorie bulk sweetener. It's got far less of a energy content than sucrose. So it is actually a billion-dollar product around the world at the moment. So xylitol production from xylose and engineered yeasts have led to near theoretical maximum yields, by which we mean pretty much all of the xylose that goes into the production can be turned into xylitol, which of course is extremely beneficial in an industrial sense. So xylose flux is entirely directed towards production of xylitol in some of these studies. A glucose limited fed batch strategy, which means that you keeping is basically what is used, this glucose limited fed batch strategy. And this leads to high productivities. So what is a fed batch strategy? Is it starts with like a normal batch fermentation where you have your media and your yeast in there, and it starts to grow, and then as the yeast grows, you add more and more of your sugar substrate, hence the name fed batch. So what they've shown is that in some cases you can get, as I said, this stoichiometric yield, complete conversion of xylose to xylitol, so 1 gram products per gram xylose. They've also used different concentrations of the sugar in the feed, and so you can get different types of titers. And it is this study that used this fed batch production that could get close to 200 grams per liter xylitol, which is very high indeed. In that strain, the ZWF1 or glucose 6-phosphate dehydrogenase and the ACS1, the acetyl coenzyme A synthetase, were overexpressed. This gene is the first step in the pentose phosphate pathway, the oxidative part of the pentose phosphate pathway, and it produces NADPH. So whereas this enzyme will produce NADH, so you've got a large pool of available NADPH and NADH, which is what your xylose reductase wants, and this means high activity for your xylose reductase in turning xylose into xylitol. So as I said, that is a product that is already in the market, and they use yeast to produce that. Something that is also very interesting is lactic acid. So lactic acid can be a precursor for bioplastics, and it is mostly made through the fermentation of lactic acid bacteria. But you can engineer yeast to make lactic acid. It requires you to express a heterologous lactate dehydrogenase gene in it to act upon pyruvate. Now, pyruvate, you'll remember, is the final product of glycolysis, and yeast is extremely good at decarboxylating pyruvate into acetaldehyde and then turning it into ethanol through an alcohol dehydrogenase. So you've got pyruvate decarboxylase, which turns pyruvate into acetaldehyde, and then an alcohol dehydrogenase, which turns it into ethanol. So anything that you want to make from pyruvate, you're basically in yeast, you're basically competing with these extremely efficient enzymes that yeast have to produce ethanol. So, and then yeast also has more than one isozyme for these pyruvate decarboxylases. It expresses a PDC1, 5, and 6, and they all compete for pyruvate. This will result in the higher production of ethanol than lactic acid, even in a yeast where you are producing a large amount of this heterologous enzyme. So you're not making the lactic acid levels that you want, you're still ending up with more ethanol. So to overcome this ethanol preference, you can delete all three of these PDCs that we saw with the different metabolic engineering methods, that apart from the heterologous gene expression, we can also knock down competing pathways. So if you knock out all of these different pyruvate decarboxylases, that sounds theoretically possible, but now you actually have a very restrictive effect on the yeast. The yeast then develops something that's called a C2 compound auxotrophy. It requires you to feed the yeast ethanol and acetate in order for the organism to thrive, which is obviously not what you want if you want to have a healthy yeast producing a large amount of your product of interest, which in this case would be lactic acid. So what they found, though, which was quite interesting, is that they introduced a Rhizopus oryzae, that is a filamentous fungus, lactate dehydrogenase in Cerevisiae, in a background strain that was also using xylose. It had the oxidoreductase xylose consumption pathway. And what they found was that it almost completely favored lactic acid production over ethanol production when you culture that strain on xylose as a carbon source. And they were able to do that without any modification of the endogenous pyruvate decarboxylases. So they took a yeast and they enabled it to use xylose with those oxidoreductase enzymes. And then they put in this lactate dehydrogenase. And what I just said was you then compete, that lactase dehydrogenase has to compete with the pyruvate decarboxylases in order to be able to use pyruvate. But what they found was when that strain, when they grew it on xylose as a carbon source, the yeast preferred to make the lactic acid over ethanol. It made almost no ethanol in those conditions. When you grew that same strain on glucose, it preferred making ethanol again. So when the same strain was cultured on glucose, it favored production of ethanol again and made 0.31 gram ethanol per gram glucose over lactic acid, which basically made up the rest. The theoretical maximum would be around 0.51 gram ethanol or lactic acid per gram glucose. So what's happening here? Why, when you grow the yeast, exactly the same yeast, on one substrate, does it prefer to make ethanol? And then when you use another substrate, that xylose, which you engineered the ability to use xylose into the yeast, why does it now produce why does it now prefer to produce this heterologous product, which is the lactic acid? And the speculation was that immediately that it comes down to the Crabtree effect. So what is the Crabtree effect? Yeast, actually, when you grow it in anaerobic conditions, it'll take glucose and it'll make ethanol. When you actually grow yeast in aerobic conditions, even when you provide oxygen to it, it will still take a large amount of the glucose and turn it into ethanol. Now, most other organisms don't do this. Most other organisms will take glucose, and if there's oxygen available, it'll prefer respiration. It'll prefer to use its electron transport chain because oxygen is available for that last step of the ETC. It can make more ATP in that way, and it's more efficient. But yeast, Saccharomyces cerevisiae, doesn't actually do that. It still turns a lot of that available carbon in the form of glucose into ethanol. It's a less efficient way of using your carbon source, but the theory is that in the niche where yeast grows, it's very competitive for free sugars, and so yeast will go and very quickly turn that sugar into ethanol, and that actually has a negative effect on all of the other organisms that will compete against it in order to use those sugars. Yeast actually makes this ethanol to kill off all its enemies, and then it can go back and actually still utilize the ethanol later on and grow on that as well, as long as oxygen is still available. So that is what is called the Crabtree effect. Even in the with the availability of oxygen, yeast will still do a fermentation reaction. It also does some respiration, but it still actually turns an appreciable quantity of that sugar into ethanol. And we saw something similar. These are colleagues of mine, or former colleagues of mine at Stellenbosch University, who engineered a yeast strain that was able to grow on xylan. So they expressed an endoxylanase and a xylosidase so that the yeast could break down xylan into xylose. And then in the same background strain, they expressed a xylose isomerase so that the yeast now had the ability to utilize the xylose as well and grow on the xylose. And so it had a complete pathway to break down xylan and utilize the free xylose sugar. And what they saw was that when they grew these strains on glucose, they saw a certain amount of cell dry mass being formed. When they grew the same strain on xylose, the strain that was expressing the xylose isomerase grew well, but actually grew to considerably higher levels than when the same strain was grown on glucose. So when we get growth of the strain on xylose, it suddenly doesn't act like the yeast normally acts on glucose. It switches from this fermentative way of growing that yeast always does because it's a Crabtree positive yeast, or that Cerevisiae always does because it's a Crabtree positive yeast, and it goes into a respiration method on this sugar that we've now enabled it to use. And what we also saw is that when we grew this strain or these strains on the two different sugars, we got much higher levels of production of our xylanase enzyme when we grew it on xylose as a carbon source compared to glucose as a carbon source. So they say that the recombinant strain displayed a complete respiratory mode, resulting in higher yeast biomass production and consequently also higher enzyme production during growth on xylose as carbohydrate source compared to growth on glucose. So that is probably something similar to what we see here in this reaction. Yeast switched away from that method where it wants to make ethanol as it normally would by using its pyruvate to produce the acetaldehyde and ethanol. And when growing on xylose, it's probably switching away from that Crabtree effect. Now, why that is interesting for us in an industry setting is the way that we would get a xylose stream in a biorefinery would be after the pretreatment step of our lignocellulosic feedstock. So we remember you have to have size reduction of your lignocellulose, and then you pre-treat it in some kind of chemical or thermal way to open up the structure of lignocellulose and allow enzymes in. And so what is generally done after this, before enzymes are added, is because these free sugars and that are produced during pre-treatment, the free sugars coming from hemicellulose hydrolysis, they will have a negative effect on your cellulases, on the action of your cellulases. So this is generally, the liquid part is generally removed after pre-treatment. So they normally press this pre-treated material and they remove this liquid part of the hydrolysate that contains the solubilized sugars that, as I said, mainly comes from hemicellulose. The solids part contains mainly your lignin and your cellulose, and the celluloses act on that, and that gives you your 6-carbon sugar mix that can be fermented. But the other sugars, those that come mainly from your hemicellulose, that's your so-called 5-carbon stream. So this contains your pentoses, your xylose, and your arabinose. And because you have them in this process as a separate stream, if we think about our biorefinery concept where we said you want to fractionate your feedstock and produce different products from these different fractions, that's actually a very handy different fraction now. So now we have a 5-carbon stream that we can actually turn into products of higher value potentially than the 6-carbon sugars, which we would turn into ethanol. So that's one option of why this, or one reason why this so-called Crabtree effect versus no Crabtree effect versus respiratory growth of yeast is very interesting in a biorefinery concept. But looking at other products that can be made, the next one is 2,3-butanediol. This has applications in plastics and fuel industries. It was shown that deletion of the pyruvate decarboxylase genes PDC1 and PDC5, along with overexpression of the oxidoreductase xylose consumption pathway and the endogenous overexpression of the endogenous 2,3-butanediol dehydrogenase gene, as well as Bacillus subtilis ALS S and ALSD genes, yielded up to 0.27 gram butanediol per gram xylose, and a final yield of 20 gram per liter in a batch fermentation. So that's the butanediol. The yeast does have a pathway towards that. We can improve that by improving this activity, either by overexpressing that native gene or expressing genes from other organisms. But again, the researchers here wanted to knock out at least part of the pyruvate decarboxylase activity of yeast. Otherwise, yeast just likes to turn pyruvate into ethanol. These engineered yeasts, though, they do not compete with the natural producers of 2,3-butanediol, such as Klebsiella pneumonia, which can reach near theoretical maximum yields of 0.5 gram of the 2,3-butanediol per gram sugar. But yeast is an organism that is relatively safe. It's not a pathogen. It has this status which stands for generally regarded as safe, GRAS status. And it has enhanced tolerance to fermentation inhibitors, as well as I've mentioned that the bacteria that we can use to produce some of these interesting products from purified sugars tend to not do well on the sugar streams that we get from hydrolyzing lignocellulosic biomass because of those inhibitors that you inherently find in pre-treated lignocellulose. Okay, the next product is 3-hydroxypropionic acid, or 3-HPA, which is a building block for biopolymers. Now, a number of these are interesting for biopolymers because bioplastics and biorubbers, remember I said these are things that we also need to think about replacing, not just the fuel, as almost all of our rubbers and plastics also come from oil. So there are two potential pathways that can be engineered to produce the 3-HPA in yeast, and they were both done in engineered xylose-consuming yeasts. And it was found that the beta-alanine and the malonyl coenzyme A pathways can both be useful. So in a strain with the oxidoreductive xylose consumption pathway, the beta-alanine pathway enhanced strain produced over 7 grams per liter 3-HPA at about 0.17 gram per gram xylose. So you'll see this is a relatively lower level than some of the previous products that we were talking about. This is past the proof of concept range, but this still needs quite a lot of optimization in order to be at industrially competitive levels for the production of 3-HPA. So in contrast, the malonyl coenzyme A pathway exhibited relatively poor production of only 0.17 grams per liter when cultured on xylose. The malonyl pathway is used for production of lots of different types of fatty acids and sterols, and generally it requires quite a lot of metabolic engineering to get those to get a product that requires precursors from those pathways. So glycolic acid has lots of applications in cosmetics, in different chemical processes, and also as a biopolymer. Yeast doesn't naturally make glycolic acid, but the glyoxylate reductase gene from Arabidopsis thaliana and others have been overexpressed in yeast, and it can turn this glyoxylate intermediate of the glyoxylate pathway into glycolic acid. So when this gene was overexpressed in yeast, the oxidoreductive xylose consumption pathway was also used in the same yeast background, and it allowed for the conversion of glyoxylate to glycolic acid from glucose. I always like this example because it's a good example of some interesting metabolic engineering in yeast, because to increase the pool of available glyoxylate, you will remember we said for metabolic engineering, you can knock out competing pathways, you can overexpress a part of the pathway, or even just one gene which encodes for a rate limiting step, and you can thereby improve the amount of your metabolite precursors and eventually of your product. So to increase the pool of available glyoxylate, malate synthase was deleted. That's this gene that would use glyoxylate for making malate, and that prevents the formation of malate, and so more glyoxylate is available to make glycolic acid. They also looked at ICL1, that's this gene over here, which produces the glyoxylate from isocitrate, and they overexpressed that to a large degree to improve the pool of glyoxylate available. And they deleted the IDP2 gene, which encodes an enzyme that converts isocitrate to alpha-ketoglutarate. And that could obviously also then decrease the pool of isocitrate, which means there'd be less isocitrate to make glyoxylate from. So that allowed the eventual production of about 0.91 grams per liter of glycolic acid. And this is, I think, interesting metabolic engineering, but again, less than a gram per liter of glycolic acid is not enough to go to industrial scale production. But it does show that there is some way to go in order to optimize the production of glycolic acid in yeast. The last example is the production of isoprenoids, specifically squalene was the one that they looked at. Squalene is a hydrocarbon and a triterpene that is a precursor for the synthesis of all plant and animal sterols. It has various types of applications. Among those are for the production of higher fuels like jet fuels and other drop-in fuels that can be used in other industries. And what they showed was that the overexpression of the HMG coenzyme A reductase with a truncate regulatory domain was found to have higher activity when cultured on xylose. So this is in the mevalonate production pathway, and when in the third year biochem course, when we spoke about the production of cholesterol, we started off by talking about this production of mevalonate from acetate. It's a very similar process in yeast up to the point of mevalonate, and we said that this gene, the HMG coenzyme A reductase, is actually the rate limiting part of that pathway. And so what they found was that when they removed or they truncated the regulatory domain of that gene, they found much higher activity in the yeast when it grows on xylose. And so you get a lot of flux through this pathway, you get a lot of mevalonate being formed. Then to produce the squalene from xylose, they again used a yeast which was already engineered with the oxidoreductive xylose pathway. And they produced the squalene by enhancing the expression of ERG10 gene and the HMG1, as I mentioned, and this allowed a high-level production of squalene, which is made from the same precursors that the yeast makes to produce all of its different type of sterols. So culturing on glucose and xylose, again, it was quite telling that there's a big difference. Culturing on glucose, you get a titer of 18.7 milligrams squalene per liter versus 150 milligrams of squalene per liter when you cultured the same strain on xylose rather than glucose. So in a fed batch fermentation, they were able to increase that to 532 milligrams of squalene from xylose. Again, an interesting proof of concept, but a lot of work needs to go into this to try and increase the levels of squalene that is produced. Okay, so to end off, just some future prospects was discussed in the article. So they say that in yeast, glucose metabolism strongly directs flux towards ethanol production. This is what I said is the Crabtree effect. Even in the availability of glucose, with the availability of oxygen, yeast will still strongly direct the flux of carbon from pyruvate to ethanol, and instead of feeding that into the tricarboxylic acid cycle as other organisms would. However, in contrast, it seems that xylose metabolism elicits a respiratory response as long as oxygen is available. So the central metabolism from xylose consumption pathways may thus be more amenable to redirection towards the desired pathways. So in other words, if you want to use acetate to make something else, or pyruvate to make something else than ethanol, or if you even want to use something that has to be made from a precursor in the mevalonate pathway, or the triterpene pathways, you are probably better off using xylose as a substrate, because then the yeast doesn't have this strong Crabtree effect where it really wants to turn anything into ethanol. So this was demonstrated that the production of acetyl coenzyme A-derived molecules such as hexadecanol, amorphidine, and squalene were enhanced when coupled with xylose metabolism compared to glucose metabolism. Yeast imposes strong regulations upon acetyl coenzyme A availability in response to glucose, and bypassing these by molecular methods, by knocking certain genes out, etc., has proven difficult. But xylose metabolism might not trigger those regulations at all. Another interesting point in growing things on xylose is it's been shown that at least some transporter proteins may undergo endocytosis and degradation in response to glucose growth. And this mechanism is also not triggered when you grow things on xylose. And that means that it's likely that the production of other value-added biomolecules which require expression of a transporter to get it outside of the cell would also be enhanced by xylose metabolism, solely because of increased secretion abilities of the cell under those conditions. And then lastly, the co-production of ethanol and high-value products may be beneficial for the economics of a biorefinery process. We've mentioned that, and so that is why it would be interesting to see if these different fractions that we can get out of the pretreatment of lignocellulosic biomass can be used to produce both a bulk fuel, a high volume, low value fuel product, as well as then a lower volume but higher value products such as these other chemicals we've spoken about. While some high-value products may not have sufficient market ability to justify being the primary product of a business, the co-production of ethanol could provide additional revenue and serve as a financial buffer against fluctuating market prices, while you also have this availability then to switch between different products that you can make in your biorefinery, making your overall process more economical.